Welcome to the DNA Extraction Virtual Laboratory. DNA is extracted from human cells for a variety of reasons. With a pure sample of DNA, you can test a newborn for a genetic disease, analyze forensic evidence, or study a gene involved in cancer. Try this virtual laboratory to perform a cheek swab and extraction DNA from human cells. If you have Flash Player enabled on your browser, you may be able to perform this activity on your own through the website at learn.genetics.utah.edu/content/labs/extraction/. And if you don't, you can just follow the virtual laboratory with me here in this video. Let's start the lab. In this experiment, you need to isolate some DNA from a human test subject. Why, you may ask, do you need human DNA? Let's find out. Scientists isolate DNA for a variety of reasons. Some of these reasons include genetic testing, body identification, and analysis of forensic evidence. DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from other proteins and other cellular contaminants. We need cells because that's where the DNA is. Inside almost every cell in our body is a nucleus, and inside each nucleus is about 2 meters of DNA. So where do we begin? First, we need to collect some cells from our test subject. We can do this through a cheek swab. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. Let's get started. The steps we will follow to purify DNA from the cheek swab are shown below. We will first collect the cheek cells, then burst the cells open to release DNA. Then separate the DNA from proteins and debris, and finally isolate concentrated DNA. Here are the materials and equipment you will need to purify DNA from the cheek cells. We have a warm water bath, a centrifuge, micropipetters, a buccal swab, lysis solution, concentrated salt solution, resuspension buffer, ethanol, and isopropyl alcohol. In order to collect the cheek cells in our virtual laboratory, we will need to drag the swab over the inside of the test subject's mouth to collect a sample. Great, we have successfully isolated some cheek cells on our buccal swab. Next, we will place the swab into an Eppendorf tube. When we look closely at the swab, we can note that it is covered with hundreds of tiny cheek cells. Inside each cheek cell is a nucleus, and inside of that nucleus is the DNA. We can clip off the tip of the buccal swab and place it into the tube. The end of the swab must be cut off so we will be able to close our tube. Using the micropipetter, we can then add some lysis solution to the tube. Lysis is a Greek word meaning to separate. We can place 1 milliliter of the lysis solution in with our DNA sample. Now we close the lid of our tube and place the tube into a warm water bath for incubation. The lysis solution that was just added contains 2 important ingredients: detergent and an enzyme called proteinase K. The detergent disrupts the cell membrane and the nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around proteins called histones. Proteinase K separates the histones from the DNA, isolating the DNA molecules. The cells have stayed in the warm water bath long enough for the DNA to be freed from the cells, and we have removed the swab from the tube. Now we will add some concentrated salt solution to our tube. The concentrated salt solution causes proteins and other cellular debris to clump together. Next, we'll place the tube into the centrifuge. Make sure to balance the centrifuge. In order to balance the centrifuge, a tube containing water is placed in the centrifuge directly opposite the tube. When we click the lid will close and the centrifuge will turn on. The centrifuge causes protein and debris to be separated from the liquid which contains the DNA. Inside the centrifuge, the tubes spin around at a high speed. The heavy clumps of protein and cellular debris sink to the bottom of the tube, while the strands of DNA remain distributed throughout the liquid. Now, we can use the micropipetter to carefully remove the top liquid which contains the DNA and place that amount into a clean tube. We want to make sure that the protein and other cellular debris located at the bottom of the tube stay behind. Great, now we're ready to isolate concentrated DNA. To do this, we will add some isopropyl alcohol to our tube. Inverting the tube several times mixes the isopropyl alcohol into your DNA solution. Because DNA is not soluble in isopropyl alcohol, it comes out of the solution. At this point, you can see the clumps of DNA with your naked eye. Next, we place the tube into the centrifuge once more and click on the lid to close it and turn it on. This time, after the sample spins in the centrifuge, the DNA will be precipitated out and will sink to the bottom of the tube. Now we can remove our tube from the centrifuge. Remember, this time the DNA is in the pellet or the precipitate that has gathered at the bottom of the tube. This is the portion we want to keep, but we want to isolate this amount from the liquid on top of it. So we remove the liquid from the DNA using a micropipette. Then we can allow the DNA to dry. After the DNA has dried, we can then redissolve the DNA in the solution of our choice. We can also store it in the freezer for many years, or move on to the next experiment, which we will do next week. This involves PCR, or polymerase chain reaction, and gel electrophoresis. But in order to do those experiments, we have to do our DNA extraction first. It might be hard to believe, but you can actually extract DNA relatively easily from just about any living thing. If you want to, you can try your hand at a DNA extraction using common household items that you can probably find in your kitchen. Let's briefly look at the steps of how you would be able to do this on your own. First, you'll need to find something that contains DNA. Since DNA is the blueprint for life, everything living contains DNA. For this experiment, we like to use green split peas, but there are lots of other DNA resources too, such as spinach, chicken liver, strawberries, and broccoli. Some sources of DNA should probably not be used, such as your family pet, Fido the dog, your little sister's big toe, or bugs you caught in the yard. Step 1. Grab a blender. Put in the blender a half cup of split peas, 1/8 teaspoon of salt, 1 cup of cold water, and blend on high for about 15 seconds. The blender separates the pea cells from one another, so now you have really thin pea soup. Next, we'll pour the thin pea soup through a strainer into another container, like a measuring cup, and add 2 tablespoons of liquid detergent and swirl to mix. Let this mixture sit for about 5 to 10 minutes. Then, pour the mixture into test tubes or other small glass containers, each about 1/3 full. Why are we adding detergent? The blending by the blender separated the pea cells, but each cell is still surrounded by a sac called the cell membrane. DNA is found inside of a second sac called the nucleus, which exists within the cell. To see the DNA, we must first break open these 2 barriers or sacs. To do this, we use detergent. Let's look at why detergent works for this purpose. Think about why you use soap to wash dishes or your hands, or to remove grease and dirt. The soap molecules and grease molecules are made up of 2 parts, heads and tails. The heads of these molecules like water and are called hydrophilic. The tails do not like water or are hydrophobic. Both soap and grease molecules will organize themselves into bubbles or spheres, with the heads facing toward the water and their tails facing inside to hide from the water. When soap comes close to grease, their similar structures cause them to combine, forming a greasy, soapy ball. A cell's membrane has 2 layers of lipid molecules called phospholipids, with proteins going through them. When detergent comes close to the cell, it captures the lipids and proteins. After adding the detergent, We will then have detergent with lipids and proteins. This breaks open the cell membrane and the membrane of the nucleus, called the nuclear membrane or nuclear envelope, and allows the DNA to escape the cell. Next, you can use enzyme powder. You can add a pinch of enzymes to each test tube and stir gently, being careful not to stir too hard. If you do stir too hard, you'll break up the DNA as well, making it harder to see. Meat tenderizers can be used as enzymes. If you can't find a tenderizer, you can also use pineapple juice or contact lens cleaning solution. Let's look at what a meat tenderizer does. In this experiment, the meat tenderizer acts as an enzyme that cuts proteins just like a pair of scissors. The DNA in the nucleus of the cell is molded, folded, and protected by proteins called histones. The meat tenderizer cuts these proteins away from the DNA, separating the DNA from these proteins or isolating the DNA. In the next step, we use alcohol to separate the DNA from the cellular debris and proteins. We can tilt the test tube and slowly pour in rubbing alcohol, which is 70 to 95% isopropyl alcohol or ethyl alcohol, into the tube down the side so that it forms a layer on top of our pea mixture. Continue pouring until you have about the same amount of alcohol in the tube as the pea mixture. Alcohol is less dense than water, so it floats on top. Look for clumps of white stringy stuff where the water and alcohol layers meet. That stringy stuff is the DNA. DNA is a long stringy molecule. The salt we added earlier in step 1 helps it stick together, so what you see are clumps of tangled DNA molecules. DNA normally stays dissolved in water, but when salty DNA comes in contact with alcohol, it becomes undissolved. This is called precipitation. The physical force of DNA clumping together as it precipitates pulls more strands along with it as it rises into the alcohol. You can then use a wooden stick or straw to collect the DNA. If you want to save your DNA, you can transfer it into a small container filled with alcohol. You have just completed a DNA extraction. Now that you've successfully extracted DNA from one source, you're ready to experiment further. Here's some more ideas you can try on your own. You can experiment with other DNA sources and find out which source gives you the most DNA. Figure out how to fairly compare them with one another. You can also experiment with different soaps and detergents, finding out if powdered soaps work as well as liquid detergents, or see if shampoo or body scrubs work as well for this purpose. You can also experiment with leaving out or changing certain steps. Now that we have told you why you need each step, find out the consequences of skipping a step or leaving one out or changing the sequence. You can also try changing of an ingredient you use. Now you're ready to answer the questions in your virtual laboratory. Thank you for watching.